Thank you.
Professor,
Uh-huh.
this is Kate, Mary.
Uh-huh.
I have a question for you. The quiz, the next quiz on Friday, on Wednesday,
Yes.
we will have a short answer or all the question multiple choice.
Both. There'll be true or false, a couple of short essays, multiple choice, multiple answer, matching. So very similar to the types of questions we've had so far
Okay, thank you.
in previous ones. Okay, if nothing else, I want to.
Sir, may I ask a question?
Yeah.
About the today assignment about the biomolecules, we should write down the answers in the boxes.
Yes, whatever works for you. Some people are writing in the boxes themselves. Other people are making a list on a separate sheet of paper, numbered from 1 to 16, and they write their answers next to the number of the cell that they're responding to. So either way is fine with me.
Thank
All right, I'm going to share my screen. And now, can you all see test 1?
Yes.
Yes.
Okay, so what I'd like to do today is go over test 1, question by question, with two objectives in mind. One is to make sure that you know the answers to the questions, but secondly, so that you know how the questions are derived. So I want to show you in the material that we've covered the source of each question on that test. And this then should be instructive for you in how to study for subsequent quizzes and tests. So starting with the first one, it's a graph. So use the attached graph to answer the following questions. What's the independent axis labeled and what is the dependent axis labeled? Independent, of course, is age. Dependent is probability of survival. And then what does the graph show for type 1 organisms? Type 1 is the top curve. And what that shows is that people, or whoever is being measured here, survived very well for a long time, through most of their life. And then at the end, crashed, died off very quickly. What does type 3 show? It shows that young members died at a young age, but then at some point the curve became asymptotic, and if they survived to middle age, those that survived lived for a long time.
Do you have headphones?
Somebody mute. Okay, was there a question about that? Okay, and where did that come from?
Excuse me, Professor.
Yeah.
Just tell the student who doesn't speak just to mute their mic because they make noise, so we didn't focus.
Yeah, I think the mic is mute now. At least I don't hear it.
There's somebody that's in a car or something. There's a lot of background noise.
Yes.
Okay, well, I don't hear that. And I don't know that, am I capable of muting them? Yeah, now I do hear that. I don't see that I have a control for it.
You should be able to mute them if you hover over their pictures or even over their name. You just click
Okay.
mute.
You're Perla?
Yeah.
If I hover over their picture, okay, first I have to find them.
At least as a host, you typically are able to. Or even on the sidebar where it lists participants.
Yeah, I got that.
Yeah. Either you can see the mute button immediately or you can click the 3 dots and have the option to mute them.
So far, everybody I've looked at is already mute.
The Mastura is not muted. Mary Ket is not muted.
Okay, I've just muted Mary Cat.
Yes, now it's gone.
Yeah, the noise went away.
Okay, so that was it. Okay. Yeah, so where did the idea for a graph question come from? Well, there were 2 points in the material we were covering on scientific method where we talked about graphs and how important they are. So that's where we got that question. The next one, it's about binomials. And we specifically talked about binomials. You can see a picture of the slide from there, which says exactly how binomials are used. When using the name in written form, genus is capitalized, species is lowercase, and they're either italicized or underlined. And when used in a paper, the first time it's used, the whole scientific name is used. The second and subsequent times, it's abbreviated. So if you look at this list, which ones are correct? Well, the first one, because it's written out, both names, and both names are italicized. The second one is not correct because it's neither italicized or underlined. The third one is not italicized. The fourth one is correct. It's abbreviated, but it's done so properly. And the sixth one is correct. It's underlined and there's a space left between the 2 words. The same with the last one. It's abbreviated, and underlined with a space between the 2 words. So that's where the question came from, and those are the correct answers to it. Now the next one, true and false. The elements of scientific inquiry include making observations and asking questions, making assumptions based on critical thinking, and communicating information. What is it?
True.
Okay, who said true?
Zach Neuschuler.
Okay.
Excuse me, teacher, could you make it full screen because I cannot clearly see?
It is a little bit small.
Yeah.
This is
Bottom right hand, you'd be able to zoom in on the actual thing.
Okay.
A little slider.
Now it's better.
Okay, you can see it.
Yes, thank you.
Okay, Zach, what did you say the answer was?
I said true.
Okay, anybody else have an opinion about this?
Professor, I'll agree. I think it's true as well.
And your name?
Marcus Kerbrock. Just from like the slide that you have there shows that they all like go back and forth.
And read the second box, the one I'm hovering over here.
Yeah, developing hypotheses and conducting investigations.
Okay, and what does the statement say?
Says that the element of scientific inquiry include making observations and asking questions, and making assumptions based on critical thinking and communicating information.
Okay. And you just said making assumptions based on critical thinking. And how does that square with this?
Because like the arrow from thinking critically leads to like developing hypothesis and conducting investigations.
Yes, that is correct. But the statement doesn't say that. What does the statement say?
Oh, it says making assumptions.
Right. And
I
so,
see.
therefore, the statement
False.
is false. That's right.
I see.
Making assumptions is not part of the scientific method of inquiry. You don't make assumptions first and then try to get evidence. You make an observation and then develop a hypothesis and conduct investigations. You don't make assumptions. So that is why that statement was graded false. Okay, the next one is about bonds, ionic, covalent, and hydrogen bonds. And that was taken directly from this slide. Ionic bonds, opposite charges attract in red. Covalent bonds are characterized by the sharing of electrons, and hydrogen bonds, electrostatic attraction. So those were the answers that were correct. Ionic bonds, opposite charges, covalent, shared, hydrogen, electrostatic attractions. Then the next question has multiple answers. Does life exist on planets elsewhere in the universe? And so I had a list of some statements that are probably true and some that probably aren't. And this came directly from this slide. The so-called Goldilocks conditions.
Pardon me, Professor. Mastur, would you please mute yourself? You have a lot of background noise coming through.
Okay.
Thank you so much.
Okay. Conditions thought likely to be required. The temperature has to be such that there is liquid water. If there is water on the planet, it has to be liquid. So the surface temperature has to be between freezing and boiling. It has to have an atmosphere and a mass that's neither too big or too small. And again, those statements are taken directly from this slide, which we discussed in class. Now the next one, standard notations are used to illustrate types of bonds. And what we're asking here is to designate the type of bond that this notation is showing. And again, this came from two different slides, actually three different slides. So sodium ion, chloride ion makes a ionic bond. This notation, a bar, either a single bar, double bar, or triple bar, any of those indicate a covalent bond. And a hydrogen bond is indicated by a dashed line. Again, this comes from the slides. Sodium ion, chloride ion yield an ionic bond. Here are illustrations of covalent bonds circled in red. And finally, a picture of a hydrogen bond between a hydrogen atom and an oxygen atom. Any questions as I go along? Is it clear what the answers are and where the questions came from. Okay, you're looking through a microscope that measures about 10, at something that measures 10 by 5 micrometers. The main color you see is green. Internal features are visible, but they're indistinct. So what are you looking at? Well, the answer is a plant cell or a chloroplast. Why?
Dylan Fengman speaking. Plant cells and chloroplasts are typically seen to be green just because of the way chloroplast works. It reflects green, and then they're typically within the 5 micrometer to 10 micrometer range.
Yeah, green is a tip-off. The fact that they're green means it's from a plant because of the chlorophyll, like you say. And the size, 10 micrometers to 5 micrometers, you can't really read this chart, but you may recall when we talked about it, that size range covers organelles and cells. And so it's the right size for either a plant cell or a chloroplast. And so then what microscope are you using? It's a light microscope. And why is that? I should put it the other way. Why wouldn't you use an electron microscope?
Because you want to see the internal features.
Yeah, but internal features can be seen with a transmission electron microscope. So what does the question tell you that tells you that it's not a transmission electron microscope?
They're indistinct.
Indistinct, right. If it was an electron microscope, you'd see them clearly, but these are indistinct. So that tells you it can't be that. It can't be a scanning scope because you can see inside the cell. So all that's left is a light microscope.
And if I may point out one other thing, electron microscopes only return colors gray scale. There's no color visible. Any color electron microscope picture you've seen has been colorized afterwards.
Yeah, yeah, again, the green color is a tip off there. Okay, fill in the blanks. Compound has the chemical formula C6H12O6. Number of atoms in this molecule is
24.
24.
24. All right, who said that?
Brian did first.
Yeah.
Okay.
Yeah.
All right, that's correct. The number of elements in the molecule is
3.
That was Caesar. Okay. And their names are?
It's carbon, hydrogen, and oxygen. This is Lisha.
Okay. We'll go to the next one. Bacterial cells are described as one of 3 different shapes, and they're shown here. This slide from a lecture. So the shapes are?
Like rod shapes?
Rod, that's right, and?
Spiral.
Spiral, helical, right, and?
And spherical.
Yeah, spherical, those 3. And this is them right here. The blue ones are spherical, the red ones are rods, and the purple ones are spheres.
Wait, Professor, I have a question. You said which ones were helical, or was that not one of them?
Yeah, helical and spiral, same thing. They're blue, the blue ones.
Couldn't those be like rope-like or no? Or do they spiral? Are they spiraling or?
Yeah, they actually do spiral.
Okay, alrighty, thank you.
But teacher, helical when the spiral get together, it makes helical?
They're the same thing.
Yeah.
Spiral and helical mean the same thing in this case.
Thank you.
Okay, fungi interact with humans in which of the following ways? Converting sunlight to chemical energy, production of pastries, yes or no?
No.
Answer is no. Don Edwards said that. Okay. Anyone agree or disagree with production of pastries? Fungi not involved.
I agree with the production of pastries because of yeast.
Because of yeast, right. And that came from this slide, picture of bread. So pastries are a form of bread. Food spoilage.
I agree.
Okay.
Agree.
Agree. There we have food spoilage. Also, decomposing litter. Same picture illustrates that. If you consider fruit, old fruit in your fridge as litter. And what about antibiotic synthesis?
I think the penicillins makes from that.
Say it again.
I think some penicillins, like antibiotic.
Oh, yes. Yeah, penicillin. And that's exactly what I'm showing here. Now, I didn't actually show you this slide in the lecture. What I showed you was a smaller slide with just the spores of penicillium. But this gives more information, which I thought you might be interested in. This is a colony. If you take these spores and put on an agar medium, it grows into a colony like this, makes a lot more spores. This is a picture of the guy that discovered penicillin, Alexander Fleming, in his lab in the 1920s. And this is typical of a microbiology lab. If you walk in one today, you'll see pretty much the same thing. Stacks and stacks of petri dishes, racks and racks of glass tubes, glass or plastic tubes. Every one of them has a microbe growing in it. This is Fleming showing a petri dish with penicillium. And I think this is interesting. This is a picture of the original plate that Fleming observed, which got him to thinking, so most microbiologists—
On the bow ties right now.
The what?
The bow ties.
Are what?
We had lost you for 5 or 10 seconds there, so I was just filling in the blanks.
Oh, okay. But anyway, Fleming sees this, and in contrast to what everyone prior to him would have done, they say, oh, damn, my bacteria, which is what he wanted, got contaminated with a fungus, and throw the plate away. Well, he didn't throw it away. Instead, he looked at this clear zone and he said, there's no bacteria growing there. There's no fungus growing there. Nothing is growing there. Why? And it turned out the reason is that the fungus is making penicillin, diffusing out into this area and inhibiting the bacteria. So it's a good example of a failure. His failure was that he got contamination and he turned it into a success. Okay, fill in the blanks. Elements represented by CHON make up approximately what percent of an organism by mass? And you're given the choice of 95, 75, 50, 25, or less than 1. What is it?
95%.
That's right. 95%. The other remaining elements needed for life make up approximately what percent?
Less than 1?
Less than 0.
Yeah, right. The answer is less than 1. And a lot of people
I understand what
had one, but they had it wrong. They had greater than one instead of less than one. So make sure you understand the use of these little carets for indicating less than one or less than or greater than. And that question came from these two slides. Now, water has both cohesive and adhesive properties. Both are important in the growth of plants. And I ask you to describe how each of these properties is used by plants in order to grow. And that came directly from this slide. Cohesion and adhesion. Cohesion is the tendency of water molecules to stick to each other, and adhesion is the tendency to stick to other things, other surfaces. So on the right here, I've got cohesion and an arrow pointing where it happens. It happens at the stomate of a leaf cell, where as water vaporizes, goes through the stomate, vaporizes, the cohesive property pulls more water up along behind it. So it contributes to the flow of water through the leaf and out into the air. Adhesion occurs in the vessels, in the xylem. The ability to stick to the sides causes capillary action, and the water wants to move up. So what I was asking here is for you to describe these two, and if you simply said stomates and xylem vessels, I gave you full credit for that. Some people had a more detailed explanation, but most of you didn't know. Which of the following statements are theories? Again, if we go to this table, contrasting hypotheses and theories. Hypothesis is a suggestion, whereas a theory is well substantiated. So if you look at these statements, matter is composed of atoms. That is well substantiated. So that's a theory. Shelf life of oranges can be extended. It's not, well, it's an observation, not well substantiated. It's not a theory. Fertilizer, not a theory. Living things are composed of cells. That's the cell theory. Hydrogen bonds are required for the structure of DNA. No one has ever observed DNA without hydrogen bonding. So that is a theory. Selenium is an element number 34. And what's asked here, so the answer to this came from several slides in the chemistry for biology section. So what's asked is, what are the number of protons? So the number of protons is equal to the atomic number, element number, so that's 34. And the number of protons is always, or almost always, equal to the number of electrons, so that is 34. Number of neutrons, how do you derive that? Well, it's given here. The atomic mass minus the atomic number is 79 minus 34, so the number of neutrons is 45. And so what's the mass of protons, neutrons, and electrons? So it's the number of protons plus the number of neutrons, which is 34 plus 45 is 79. So each of these numbers came from a list that you were given. So are there any questions about why this question was asked or the answers? So if you studied these slides from the chemistry section, these answers should be obvious. And I should say that you should go over these again, because these questions or similar questions will probably come up in subsequent tests or quizzes. Okay, match the level of organization with its description. Well, this came from a worksheet, actually, where you were supposed to have gone into your textbook, pages 9 to 12, and filled in this table. And if you did, you came up with an organelle is number 3, a membrane structure within a cell. An ecosystem is number 6, collection of organisms. An atom is number 8, smallest unit of matter. An organ, number 1, a collection of tissues for a specific purpose. Molecule, 7, structure composed of 2 or more atoms. And a cell, number 4, smallest living thing. So any questions about that? Or where the question was, how the question was derived? Okay, match the terms below to the list. So what are these various elements used for in living things? Well,
Can I say feature some of them?
Yeah.
Calcium is need for bones and teeth, and also for muscle contraction. Magnesium is an enzyme cofactor, and iron is for carrying oxygen.
Right.
In all these, copper requires for hemoglobin production, and potassium electrical balancing of nerve cells, and also on the heart function.
Yeah. So these are all correct statements.
Yeah.
And where did we get them? It was from this slide that was taken directly from one of the lectures. Okay, water is found in 3 different states. For each state, indicate the level of hydrogen bonding among water molecules. And that question is taken from this slide, which shows that when water is solid, ice, all of the water molecules are hydrogen bonded to other water molecules. In liquid, it's some of each. Some are bonded, some are not. Whereas in gas, in the gaseous form of water, there are no hydrogen bonds. They're all broken. And so that's what this question was asking. Liquid is C, partial bonding. Gas is E, no hydrogen bonding. And ice is A, complete hydrogen bonding of all molecules. Right, then this is an essay question. I wanted you to think about the discussions we've had about life elsewhere in the universe. And so what I pointed out in asking the question is that intelligent life on Earth is less than 0.2% of life's history on Earth. And then make the assumption that the same thing either has happened or is happening on other planets in the universe. What's the likelihood that they would find us or we would find them? And based on the discussions we've had and the reasoning, the likelihood would be low. And it's derived primarily from this illustration, billions of years and life. So life has existed for about 3.5 billion years on this planet. Intelligent life is way down here, almost non-existent in terms of time. So that means that life on this planet has been unintelligent most of the time. Now, if the same thing is happening on other planets, then at any point in time, life on another planet is likely to be primitive. Consequently, if it's primitive, it can't find us, so we have to find it. And what are the chances of that? Well, very low, because the distances are so great. If we have to actually travel to another planet and dig up primitive forms of life there, the distances are so great that it would take millions of years to do that. So likelihood of finding life on another planet, even though life on other planets likelihood is high, the likelihood of finding it by us is relatively low. Okay, water has unique properties that make life possible, and one of these is that ice floats. So the question was, ice floats because we have several different statements here, and this is taken from the fact that that hydrogen bonding causes ice to float, taken from this slide. So the correct statement is that the density of the liquid state of water is greater than its solid state. And that's what you see here. Well, this one is very similar to a previous question. Atomic mass is defined as the sum of the protons plus the number of neutrons. Which of the following are lacking in animal cells?
The cell wall and the large vacuole.
Okay, okay. Cell walls, large vacuole, and what else?
The chloroplast, right?
Chloroplast, right. And that came directly from this slide where it says not in animals. These three are not in animals. Okay, here's another question about carbon, hydrogen, oxygen, and nitrogen. And it simply asked you to identify the words. That was fairly easy. It came directly from this chart. Choose the statements that correctly describe a virus. Well, we had a discussion about viruses. There are viruses that infect prokaryotic and eukaryotic cells. Different viruses infect different organisms. They're very small compared to the cell they invade. And the reproductive process differs in that they have to take over the cellular machinery of their host cell. So if you go back to the possible answers, first, viruses can infect plants and animals, but not bacteria. Well, that's not true because
Because it's infected bacteria.
right, this is true. It can infect anything. Viruses insert their own reproductive machinery into a cell. That's not true because viruses rely on the cell's reproductive machinery. They inject genetic information into the target cells in the form of DNA and RNA, which programs the target cell's machinery to make new viruses. And viruses are smaller than the cell they infect. They can't be larger. So those are the answers to those questions. Then we have this one, why are we not just one big cell? And that came from a discussion we had in class surrounding this slide. And the short answers are given here in the lower part of the slide. We have many cells to facilitate transport of materials and communication. In order to have cell specialization, and for injury repair. If we had one big cell, that cell got injured, then the whole organism is dead. Whereas with many cells, you can injure a few of them and the organism can recover because there are still lots of other cells left. So those are the answers that I was looking for this question. It was an essay. Well, true or false, organisms in the domain archaea are unicellular, found in extreme environments, and are of major interest because of their ability to cause human diseases. True or false?
False.
Laith? Yes, it's false because one of the characteristics of archaea is that none is known to be a pathogen. Okay, match the terms with the most correct term on the right. A phylogenetic tree is relationships based on ribosomal RNA sequences. Again, we're getting these questions and answers from these slides that were shown in lecture. Morphology is a relationship based on visual similarities. And these are two key words, homologous structure and analogous. Homologous structures are, for example, bird wings versus bat wings. They have a common evolutionary origin, whereas an analogous structure, bird wings versus insect wings, they have similar functions, but they're derived evolutionarily, they're derived differently. And a domain is the broadest level on which organizations are classified. So again, based on the information in these slides showing that ribosomal RNA is useful for classifying organisms. And this shows the difference between analogous functions and homologous functions. So bat wings and bird wings are homologous. They're derived from the same source evolutionarily, whereas insect wings are analogous. They have similar functions, but different origins. And this shows the three domains, which are the broadest categories we have for classifying organisms. And then there was an essay question about being an intern in a lab and your boss asks you to, it says it wants clear pictures of the internal fine structure of an organism's Golgi apparatus. Tells you to find a place to have this done. The question is, what subdepartment would you call to schedule the imaging appointment? Based on the information here, the boss wants clear pictures of the internal fine structure. So what department would be called for internal fine structure?
The second one, teacher? Microscopic.
Say that again.
Microscopic should be directed by microscope.
They're all microscopy.
Scanning. No, a scanning electron.
Scanning? Anyone else have a thought?
Or transmission electron microscope.
I'm thinking it's transmission electron.
Yeah, transmission electron. And why? Why would you choose transmission?
Scanning electron microscopy only does the outside surface, whereas transmission does through the entire thing. So you're able to see the internal structures.
Right. And you wouldn't choose light microscopy because it's not powerful enough to see internal fine structure of something that small, 35 micrometers. Okay. And that's based on this type of slides. True or false, Protista is a kingdom of eukarya that contains all forms of life that don't fit in any other kingdom. Is that true or false?
True.
Who said that?
Marcus Carebrook.
Okay. Yeah, it's true. And I guess I don't have the slide that came from, but yeah, it's sort of a garbage bag, grab bag of everything that doesn't fit in a nice clean category. It's called Protista. And it is a kingdom, not a domain. Okay, so my point in going over all these questions in detail was, as I said at the beginning, twofold. One, so that you know the answers, and the other is you know how the questions were derived. So anyone have comments or questions overall?
Could we have it so that way we can see what questions we actually got correct on our exams? Because right now we're just able to see our final score.
You want a copy of this sheet? This is a Word document. You're asking for that?
That would be nice, but also in Canvas itself, we're only able to see like the final score that we got. We're not able to see which questions we got right, which ones we got wrong.
Oh.
Yeah, it says the results are hidden.
Yeah, it would be nice to have the results.
Okay, well, I thought Canvas allowed you to see that. Well, I'll certainly, I'll check and see about Canvas, but also I will send you this Word document, which I've just shown you.
Wait,
Nice.
so have the written questions already been graded too then?
Are you asking, has test one been graded?
Like the written questions, because when you first take it, it says that we have to wait for the written questions to be graded.
Yeah, they all have been.
Oh,
If you
damn.
look at your score, it should show the complete grade. I mean, the grading of all the questions. Okay, I'll send this to you so you can use it. Yeah, it's a good idea to use this as a study guide for future quizzes. All right, anything else? If not, I will stop sharing this.
Did have a question on the oh god, my brain just stopped. The question about the I'll ask later. Never mind. It's fine.
Okay. All right, I'd like to move on now to, I'm going to have to go fast to the topic for today. More DNA and RNA. Everybody sees that.
Yes.
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 All right, and a little review. DNA makes up the genome. RNA is the intermediary that makes proteins from the information in DNA. Now, I'd like to stop or digress, I guess is a better word for it, and point out, first of all, a momentous discovery, one of the most significant in the history of science, and also a bit about how science can be done. So going back, 1951, James Watson, Francis Crick, and Rosalind Franklin were involved in the discovery of the structure of DNA. And here is Crick in later life and Watson. Crick is now dead, Watson still alive. Rosalind Franklin died very shortly after the discovery of the structure of DNA. Now, I want you to think about the state of genetics before the 1950s. Most scientists thought the genes were made of proteins. And that made sense because they knew that there are thousands of proteins in an organism, and organisms can have thousands of traits. So the hypothesis was that each heritable trait is controlled by a specific protein. And a huge amount of effort was made trying to prove that was true, and of course it never was. But when you think about it, what were the possibilities? If you were a scientist, didn't have any idea what the chemical nature of genes were, and proteins are a possibility, what were the other possibilities? We've talked about biomolecules, macromolecules. What are they?
They are amino acids, proteins, lipid, and carbohydrates.
Carbohydrates.
Yeah.
And lipids.
Lipids.
And
amino acids.
Nucleic acids.
Nucleic acids, yeah.
Okay, so those are our four major categories. And so scientists said, well, we can rule out carbohydrates because there's not enough variation. Carbohydrates are just long chains of sugar monomers, no information there. Ruled out lipids for the same reason, and ruled out DNA because, it says on the bottom left here, it was widely thought to be a boring molecule composed of only 4 variable parts. They knew that. And they were claimed to be present in identical quantities, which turned out to be wrong. But at that time, no one imagined that genes are encoded, that each gene has a specific code based on the 4 variable parts. But in 1943, a man named Oswald Avery demonstrated that a substance that he got from virulent bacteria could convert avirulent bacteria into virulent ones. And that substance was DNA. So think of how Avery did this experiment. He had cells of bacteria that were virulent, other cells that were avirulent. So he made an extract of the virulent cells, took everything out of them, small molecules, large molecules, everything. And then he purified the various different components, threw away all the small molecules, they didn't count, and he ended up with a fraction containing protein and a fraction containing DNA, another fraction containing RNA. Well, it turned out that the only substance that converted avirulent to virulent was DNA, which was really a striking result, but almost nobody believed it. Why? And consider how Oswald did this. He started with very crude cell extract and ended up with a fraction that he purified, and he said that fraction is DNA. Well, people didn't believe it because they said, it can't be DNA. And his DNA fraction was contaminated with protein. And there was no way to prove that that wasn't true. So no one believed it, or almost no one believed Oswald. Then in 1950, a man named Erwin Chargaff showed that if you looked at the percentages of A's and T's and G's and C's, A always equaled T, 30%, and G always equaled C, 19%. Now this was, again, it was a profound result, very striking, but nobody knew what to make of it until Watson, Crick, and Franklin came along. So in 1951, Watson and Crick were at the Cavendish lab, and Franklin was at King's College. And Watson and Crick knew that Rosalind Franklin was an expert in X-ray crystallography. It was very difficult in those days to make a crystal of DNA, but she knew how to do it. And she knew how to shine X-ray beams on it and get a diffraction, which you see in this picture here. Well, Watson happened to know that regular substances like crystals diffract X-rays in characteristic patterns. And you can see this is a characteristic pattern. So he and Crick went to King's College to talk to Franklin, asked to look at her pictures, and she showed them, among others, she showed them this one. Well, Watson knew immediately that this picture shows a helical molecule with regular repeats. So he sort of knew the structure just by looking at that X-ray picture. He didn't know how many strands there were. Could have been 2, 3, or 4. And he discovered the number of strands by building models. And there's a video showing, this is Watson in his old age, showing how he made cardboard cutouts representing T's and A's and G's and C's. And he just moved them around on a tabletop and showed how they could match up. And based on his model building, he came up with the structure of DNA, which you can see here is a metal construction. And you can see even in 1951, they knew it was a helix. And so one of the points that I want to make here beyond the historical interest in it is how Watson and Crick made a momentous discovery. They didn't do it in the ordinary way. What they left out was this box: developing hypotheses and conducting investigations. They didn't have a hypothesis for what the structure of DNA was. And they didn't really conduct investigations. They looked at one picture from Rosalind Franklin, and they made cardboard cutouts and flipped them around on a tabletop. They didn't actually do experiments. And yet they were able to deduce the structure of DNA. And the point is that this approach is not typical of the way scientific discoveries are usually made. It worked for them, and it had a terrific impact on science and on biology. And what was important is that when other people actually did chemical analyses, actually did investigations, they confirmed that Watson and Crick's model was right. So whatever works. Any questions about how this came about? Any of you who are interested can read. Watson wrote a book called The Double Helix, which is a very interesting book to read because it describes all of this. Okay, what are the 4 bases? A's and T's, G's and C's. Now this is a picture that you've seen before, and what I want to emphasize here is, what is a nucleotide? Lower right-hand corner shows one DNA nucleotide. A nucleotide is made of a phosphate group, a 5-carbon sugar, and a base. The only variable is the base, and there are only 4 possibilities in DNA: G or C or A or T. The phosphates and sugars are always the same. They just repeat over and over and over. And the A's and T's and G's and C's repeat over and over. And it's the variation in these bases that establishes the code for specific genes. You've seen this before. DNA is a double helix. The force that holds the 2 strands together is what? What are these bars representing A and T?
Hydrogen bond?
Hydrogen bonds, right. Now, this just shows how A's always match with T's and G's always match with C's. But importantly, I want you to notice the blue bars versus the red bars. The hydrogen bonds between A's and T's, there are 2. Between G's and C's, there are 3. So remember that. Now, what is a gene? And this is something that you need to know, you need to remember this, because a gene is just a section of DNA that codes for a protein. And these red letters represent a section of DNA that encode a protein. The sequence of nucleotides for each gene is unique. The nucleotides are read in groups of 3 letters, and we'll talk more about this next time. And it's estimated now, we know the complete sequence of the human genome. We know that it contains about 20,000 protein coding genes. That makes up less than 2% of the total DNA. So all of the characteristics that we have are encoded by less than 2% of our DNA. And think about that. Every cell in our body, almost every cell, has a complete genome. So we're carrying around in every cell 98% of our DNA that doesn't code for anything. So that is a big mystery, and it's slowly getting worked out. And it turns out that all of this non-coding DNA, non-coding does not mean non-functional. So some, or perhaps most of it, is involved in gene regulation. And it's carried around over evolutionary time because natural selection has selected for it. In other words, it's being used for something, otherwise it wouldn't exist anymore. So this 98% of non-coding DNA is used for something, and it's thought to be largely for gene regulation, transcription factors, introns, enhancers, silencers, and we'll talk more about that later. Genes are located on chromosomes, and this is called a karyogram of a human female. Humans have 22 pairs of what are called autosomes, body chromosomes, and one pair of sex chromosomes, XX in female, XY in male. We'll talk more about karyograms and what they can tell us in a subsequent lecture. But this karyogram is just a picture of chromosomes. And we will come back to cystic fibrosis soon. And we'll talk about the CFTR gene, which is entirely responsible for cystic fibrosis. The normal function of this gene is it encodes a protein. It goes to the membrane of epithelial cells and moves chloride ions across membranes. Chloride ions. Does anyone remember what a chloride ion is? Think of salt. What happens to chloride in salt?
A chloride ion has an extra electron.
Right, that's what makes it an ion. So it's a negatively charged ion because it picked up an electron. Well, chromosomes are located on genes, and genes, every gene has a centromere. And when we talk about cell division, we'll talk about the function of a centromere. But on either side of the centromere is a length of chromosome, which is called either the long arm, Q, or the short arm, P. And then if you stain chromosomes with various dyes, they exhibit a banding pattern. And every gene has a chromosomal location based on which chromosome it is, which arm, and which band. And so you'll see codes like this, 7q31.2. Well, what does that mean? This is actually the location of the CFTR gene. So 7 means that it's on chromosome 7. That's this. Q means it's on the long arm, Q. 3 means it's in region 3. It's this region here. 1, band 1, and 0.2, subband 2. So this is how geneticists indicate where on any chromosome a gene is. So even now that we know the sequence, a complete sequence of every chromosome, chromosomal locations are still used. This code is still used. So be able to indicate for purposes of future tests how to interpret a chromosomal location code. Chromosome 7, one we just looked at, contains over 150 million base pairs. The CFTR gene has 257,000 of these, 26 introns, 27 exons, and we'll talk about those later. And I've used up all the time. So Scott, would you like to say something about the workshop coming up?
Yes, I have a workshop coming up about 17 minutes from now. Remember, I start it only a minute or two early, if at all. Normally, I'll start it right on time. So if you get there early and it hasn't started yet, please come back, try again, because I will open it up. I am having one today. Good chance for you to come in, ask questions about the exam, although you shouldn't have too many questions at this point because you got to see exactly where the questions came from. And great time to go over biomolecules and the genes and all this new material. So hope to see some of you there.
Okay, please go. And I will see you on Wednesday And Wednesday, the class will be 60 minutes instead of 75 because you'll take 15 minutes for the quiz. And the quiz will be available all day, as usual, from midnight to midnight. Okay,
Professor,
yeah,
could we get the slides put up on Canvas so we're able to take a look at them?
they should be in Canvas already. If they're not, I'll put them in Canvas immediately. So I'll make sure that the slide set and the answers to the questions of test one, I'll put them both in Canvas.
Okay, cool. Because I'm only able to see up to week 4, the week 4 module, so no week 5 module.
Yeah, the biomolecules is the last slide uploaded.
Okay, then I'll get this one up today. Okay, anything else? No, then head over to Scott's workshop. I'll see you Wednesday.
Have a great day, Professor.
Okay.
Have a good one.
You too.
Thank you, teacher. Have a great